Well, what I'd like to do is start in a little bit early and talk a little bit more quickly than average because I've got to make up for some slides that I did not cover at the end of last week's lecture on color vision. And so I'd like to finish chapter 4, and you can see I'm going to talk about color opponency shortly. And this is a slide that we already looked at this past Thursday. And then we'll launch into chapter 5. So without further ado, allow me to remind you that one thing that happens with color vision is a comparison of light levels found in different regions of the visible spectrum. And the way by which color signals or signals from photoreceptors are compared to one another is known as color opponency. And this is a theory that is based on perceptual phenomena as well as on physiological and psychophysical measurements. So perceptually, there appear to be 4 primary hues. And in addition to the red, green, and blue that we talked about, well, there's also yellow. And you have to note that, yes, while it is possible to see red and yellow things, namely orange, it's not really possible to see things that are red and green in the same place at the same time. So they're opposed to one another, those colors, as are blue and yellow. So then we looked briefly at unique hues, and these are unique red, green, blue, and yellow. And there's unique blue, which appears neither reddish nor greenish. Unique yellow, ditto. Unique green appears neither yellowish nor bluish. And finally, unique red is that perfect red, neither yellowish nor bluish. And it turns out that that is not a color that you can generate by looking at lights in the visible spectrum. Rather, you've got to add up a violet light and an orange light. It turns out that the blue and yellow in those 2 lights will cancel one another to give you a nice unique red if you do it just right. So, hue cancellation is the sort of experiment that Hurwitz and Jameson performed in the 1950s to sort of put color opponent theory on a quantitative footing. So, for example, they noted that, well, starting in the long wavelength region of the spectrum where things are somewhat orangish, all the way through, I don't know, what is this, about 500 nanometers or so, all of these lights appear to have some yellow in them. And they wanted a way to figure out how much yellow there is in the lights in this region of the spectrum. So what they did is they canceled the yellowness in all of those lights using a fixed blue light. So they chose a fixed blue light, and they asked, how much of that blue light do I have to add to light of, well, let's choose this wavelength right here. That wavelength might be 570 nanometers or so, and they said, okay, how much of that blue light do I've got to add to light of this wavelength to cancel it? And they found, aha, we need a certain amount of blue light, and we're going to plot that number. If we now ask how much blue light do we need to add to this light out here to cancel its yellowness, it turns out that there is less of that blue light needed to cancel the yellowness. And you can do that operation, how much blue light is needed to cancel the yellowness for this entire range of the spectrum, and you'll get the measurements indicated by the yellow squares. You can then do the following cancellation experiment for bluish regions of the spectrum, and you can say, okay, how much of a fixed yellow light do I need to add to lights from this region of the spectrum to eliminate the blueness that I see? So if I were to take something at 450 nanometers, I'd say, well, there's a lot of blue there. How can I measure how much blue there is? Well, I take a fixed wavelength of yellow, say this one right here, and I ask how much of this guy do I need to add to that guy to cancel the blueness? Just get rid of it. Not too much, just the right amount. And you'll say, well, I need to add that much. If I now ask the same question, how much yellow light do I need to add to this particular light? The answer is far less. So what you are able to do with hue cancellation is effectively measure objectively how strong a particular color's appearance is as you vary wavelength across the visible spectrum. And you don't need to confine yourself to blues and yellows. You can also ask about reds and greens. So you can say, well, in this region of the spectrum here, more or less in the middle, lights have some green in their appearance, and that's very strong, and maybe the 530 nanometer range, and you can say, well, okay, let's figure out how much green there is in all of these lights. How do you do it? You pick a fixed reddish wavelength, maybe, I don't know, let's say 660. I just made that number up. This is going to give us some red, and I'm going to say, okay, how much of that particular wavelength do I need to add to this guy right here to cancel the greenness? It's going to be a large amount because there's a lot of green here. If I do the same thing for that wavelength, now there's less green. I need less of that red light to cancel its greenness. If I do the same thing here, I need less of that red light to cancel its greenness. And I plot the amount of that red light needed to cancel the greenness, and this will be the result. Finally, we know that lights at either end of the spectrum look red. So the lights out here look red, and we also know, and this is not a very good picture, that lights at this end of the spectrum, the short wavelength end of the spectrum, tend to look purple and violet. And if you think about what violet looks like, that's a combination of blue and red. So there's definitely some redness at the shortest wavelengths also. And if we look at the hue cancellation functions, we'll see, yes, there's some red light, red appearance here at the short wavelengths, and some red appearance here at the long wavelengths. So does that make sense? So these are Hurwitz and Jamieson's color opponent functions, and that's how they measured them. And what we want to do is understand where they come from. And the current thinking is that cone photoreceptor responses are compared to produce a sort of second stage of color processing, and it is the responses of the second stage color sensitive cells that are responsible for color opponent appearance. So let's consider red versus green, this particular pair of opponent colors. What we're going to do is ask, well, what's a reasonable way to combine the inputs of the short, medium, and long wavelength sensitive cones, S, M, and L? And we know that short wavelength lights do produce some sensation of red. The medium wavelengths of light typically do not give rise to red, rather to green, but the long wavelengths of light again give rise to red. So perhaps you might hypothesize that short wavelength sensitive cones contribute positively to redness. Long wavelength sensitive cones contribute positively to redness. And for this cell, medium wavelength sensitive cones, well, they inhibit that cell, and in so doing cause greenness. And that is more or less the standard account. If this cell, represented by the plus sign, which combines excitatory input from short wavelength sensitive and long wavelength sensitive cones, and inhibitory input from medium wavelength sensitive cones, if this cell is excited, namely if it has a positive response, then things will end up looking red. If this same cell has a negative response, that means, well, it must be strongly inhibited by the M cones, and things will end up looking green. So the response of that cell, if it's positive, means red. If it's negative, it means green. And if it is 0, it will look neither red nor green. Does that make sense? That is a cell responsible for red and greenness. What about a cell responsible for yellow versus blue? Well, this is the standard wiring diagram for that. If we want a cell that is excited by the short wavelength sensitive photoreceptors, then we'll have a little plus sign here. And the excitation of this cell will lead to the appearance of blueness. Opposed to this short wavelength sensitive cone input is a combination of medium and long wavelength sensitive cone inputs. And these inputs act in an inhibitory fashion. You see this minus sign right here. So it ends up being a short wavelength sensitive input acting in an excitatory fashion, a medium and long wavelength sensitive input acting in an inhibitory fashion. And this combination here is thought to effectively present a yellow inhibitory input. And so if we have a negative response from this cell represented by the plus sign, then we'll end up seeing yellow. And once again, if this cell doesn't respond at all, that means that, well, it's not excited, so we won't end up seeing blue. It's not inhibited, so we won't end up seeing yellow. We'll see neither blue nor yellow as a result. So if we look at the response of a blue-yellow cell in monkey, and you can find these in retina, in LGN, and several visual areas, you can find a particular cell that's excited by short wavelength lights. And here we have lights varying in wavelength from 400 to 700 nanometers, and we see that, yes, the firing rate is higher at the shorter wavelengths, so it's excited, and it's also inhibited by longer wavelength lights. So its firing rate is now less than the spontaneous firing rate, which is the firing rate that you would measure in the absence of any stimulation whatsoever. So this is effectively the zero balance point. This is excitation at short wavelengths, inhibition at longer wavelengths. So this would be a blue-yellow cell with effectively the ability to compare the amount of light at short wavelengths to the amount of light at this medium and long wavelength zone. So you might be asking, what about black and white? Because we need 3 dimensions to account for color vision. And the simplest way to think about black and white sensitivity is to say that there are cells that simply add up the inputs from short, medium, and long wavelength sensitive cones. And the basic notion is that, well, the greater the response, the brighter something will appear. The less the response, well, the darker it will appear. And so that would be a 3rd channel in the color opponent theory. And in fact, it's not really a color channel, and it's not really an opponent channel the way the other 2 were. Anyway, that's probably the simplest version for a white-black channel. Now, color opponency. Is there any perceptual evidence beyond the things that we talked about for color opponency? And it turns out there are some illusions that provide really nice evidence for color opponency. So this is a color version of a stimulus that we looked at earlier for simultaneous contrast. In that stimulus, we had 2 gray disks, and they were surrounded by white and black placards, respectively, and the grays did not look the same. So now we're surrounding them by this sort of reddish and this greenish on either side. Do they look the same, those 2 grays? Most people would say they look sort of the same. Or if they do differ, it's not by very much. But if you sort of stare right here, right at the border, right in the middle, without moving your eyes, I think you'll be able to see a little bit more easily that there is an effect. Do people see a difference now between those 2 grays? Good. And there is a spatial comparison process going on. And what happens is that, well, when we see this gray surrounded by the red, we end up emphasizing the difference. And the difference here is a color opponent one. So we end up seeing the opposite of red. What is the opposite of that red? It's sort of a greenish color. Likewise, it's a spatial comparison going on here. The gray is compared to green. So we're going to emphasize the difference, and this is a color difference. And so we're going to emphasize the opposite of green, which is red. So that's simultaneous contrast in the color version. And here are Chevreul stripes now for saturation. Recall that saturation is the dimension that's effectively telling you how strong coloration is. A gray color would be no saturation whatsoever, zero saturation. A highly colored thing, well, that would be a high amount of saturation. So here we're going from low saturation to high saturation. Each of these vertical bars is uniformly saturated. And yet, if we were to look at one of these bars, say this here, I think everybody would agree that it appears to be less saturated on its right side and more saturated on its left side. Do people agree that there is a variation in perceived saturation? Yes. And that again indicates that there is a process of spatial comparison going on in the visual system. Saturation levels are being compared at neighboring locations in space. And if there's an edge, such as the edge between adjacent stripes, then the differences that are found will be emphasized. Finally, successive color contrast is probably everybody's favorite. And this is something that you'll see by way of demonstration. You've got to actually do this. So we're going to stare at the black dot on the flag for 30 seconds. And when I say stare at that black dot, that doesn't mean look at it, then look over here, then look here, and then look there, and then look at your friend. It means stare right there without moving your eyes for 30 seconds. So here we go. 1, 2, 3, 4. "26, 27, 28, 29, 30. Look at that black spot on the right. How many people get a very nice flag there? Good. Okay, so that is an example of successive color contrast. First thing to note is that the word successive refers to the fact that, well, you did one thing at time A, and then you got your effect at some other time B, namely time B is later, after you looked at the initial stimulus. So it's one happens after the other. That's successive color contrast. Second, what is the color that appears to be the opposite of yellow? Blue. What is the color that is the opposite of green? Red. What is the color that is the opposite of black? White. Okay, so color opponent theory comes to life when you start looking at things like this. In any event, that is what I wanted to say at the end of last week's lecture, and I'd like to move on now to Chapter 5. So, give me a moment and we'll do that. Close this one, open this. Okay, now we're starting in on Chapter 5. Chapter 5 covers a lot of material on visual perception, and we're going to cover all that material. It starts with an examination of how the visual system processes form or shape information, and it starts by noting that there are many types of cell in the visual system. And if you were to look in primary visual cortex, for example, area V1, you would find cells called simple cells. And it turns out that simple cells are responsive to bars or edges that have particular orientations. Now, have I mentioned these guys before? Orientation selective cells? Well, let's suppose we have a cell with a receptive field that is selective for a vertically oriented bar. Then its receptive field might have the properties diagrammed here on the right. There would be a central region where if you shine a light, the cell will be excited, and there will be two flanking regions on either side where if you shine a light, the cell's response will be inhibited. And the excitation is marked by the plus signs, the inhibition is marked by the minus signs. And this is a receptive field, sort of a modeled receptive field of a simple cell responsive to a vertical bar of light. Now, you might ask, well, where does the receptive field, where does that property, where does that orientation selectivity come from? Well, we already know that there are center surround cells, and if you shine a light in the center, it might be excitatory. If you shine a light in the surround, it might be inhibitory. It turns out that we have center surround cells looking at any direction in visual space. So I'm now looking at you, and I know I have lots of center surround cells looking at you and in your neighborhood, but there are also center surround cells in my visual system that are looking all over the place. You name a direction, and I've got center surround cells looking there, even if I personally am not paying attention. You have those center surround cells too. So what you've got to imagine is you take center surround cells that are looking at three adjacent locations, in this case one above the other, and you simply add up their inputs. And what you would have is a new responsivity, a new selectivity, and this would be a selectivity for a vertical bar. Okay, so the notion then is that simple cells, which are orientation selective, combine the inputs from several neighboring center surround cells. If you want vertical selectivity, then you want to have center surround cells that are sort of looking at regions of space, you know, one on top of the other. If you want to have a horizontally selective cell, then you want to have inputs from center surround cells that are looking at regions of space sort of right next to one another, varying from left to right. So notice that the bar of light, for it to respond best, really should be placed in the middle of this receptive field where those plus signs are. If you have a bar of light and you place it on the left side, well, that cell won't like that at all. It will be inhibited. If you put it over here on the right, also it will be inhibited. Really, this cell is very positionally selective as well. You've got to put the bar of light in the middle of the receptive field. And it turns out that this kind of cell is linear in its response to light modulations. And what that really means is that the response of that cell is basically a sum of its responses to the lights arriving in its visual field. But it also means that it acts in a fairly simple way that's easy to understand. We have an example of something different coming right up. Okay, at each direction in the visual field, we have neurons in V1 of many sizes and of all orientation selectivities. So if I now look at you again, and I consider different parts of the visual field, not only do I have center surround cells looking at each part of the visual field, I also got these neurons that are sensitive to vertical edges and bars. I've got neurons sensitive to horizontal edges and bars, and I've got all the different diagonals as well. And you have these too. So basically, visual image change is analyzed according to both scale and orientation in all directions of the visual field. And so here we have an example. We have four orientations illustrated here. Horizontal, 45 degrees, vertical, and 135 degrees. It turns out at any location in the visual field, orientation selectivity varies more finely even than 0, 45, 90, and 135. If you pick some angle like 122 or something, there is likely to be a neuron that's really, really close to just that number. So there's a large number of neurons looking at each region of the visual field with a different orientation selectivity and size. So in this picture, you can see we've got some horizontal things going on here, these steps, some more horizontal things up at the top, and then we got these slanted lines here. And so it turns out that if we were to look at the world through cells that are selective for a particular orientation, well, that's very similar to filtering an image in a way that retains what's happening at a particular orientation and throws away everything else. So this particular filter is most sensitive to edges and bars that are horizontal. And if we look through this filter, we see that these steps over on the left come through very well. Those large edges at the top right come through very well. These diagonal things, though, come through relatively poorly. Here's an example at 135 degrees of orientation. We're now filtering things to emphasize diagonal stuff, and you can see that these diagonal lines on the floor here come through very well. So effectively, each set of orientation selective cells is finding a different set of features across the entire visual field. Now, actual receptive fields do not have cookie cutter shapes. For center surround cells, it's been this little disc in the center and then the annulus surrounding it. In fact, that's not quite accurate. It's a little bit too cut and dry. Receptive fields typically have smoother profiles. And for center surround units, these resemble more closely normal distributions or Gaussian functions. So this is an example of a Gaussian function. This is another example of a Gaussian function. And if these do not ring a bell, well, these are the normal distributions you may have encountered when you looked at statistics. How many people know about normal distributions? Good. Well, these are the same things. What we can do is we can make them taller or higher, or to have greater sensitivity, or we can make them have lesser sensitivity. We can also make them narrower or broader, and this one is narrower and that one is broader. And it turns out that the standard model for a center surround receptive field has an excitatory center given by one Gaussian function, an inhibitory surround given by a second Gaussian function that is broader, and you take one and subtract the other to get the result, which is called a difference of Gaussians. And you can see that there is a negative lobe on the left, a negative lobe on the right, and this sort of positive zone in the center. When you think about it in a one-dimensional fashion, the actual model is two-dimensional and is shown here using brightness. So the excitatory center is shown using a white that is brighter than the gray background, and the inhibitory surround is shown by a darker gray than that of the background. So effectively, we're coding the receptive field excitation and inhibition by different values of gray. In any event, that's a better model of a center surround receptive field. Likewise, here is a better model of a simple cell receptive field. And what you do is say, well, no, it's not really this. It's more the idea that you multiply one of these smooth Gaussian functions by a sinusoid. This turns out to be a sinusoidal bar pattern. And if you do this multiplication the right way, you'll effectively get a smooth look, a smooth window onto this bar pattern. And that smoothed bar pattern will in fact be a better model of the receptive field than this thing up here. So these are known as Gabor functions. And the receptive field profiles of the orientationally selective simple cells in primary visual cortex are often fit well by such Gabor functions. So, now, so much for simple cells. It gets worse. What about complex cells? Well, they're more complex. Should we just leave it at that? No. What you do to get a complex cell, it is believed, is to simply take the inputs from neighboring simple cells, and instead of adding them up, you combine them in a nonlinear way that results in the following. The first thing is that you have complex cells, well, they are orientation selective, and what we have here is a complex cell that is selective for a bright vertical bar. But the thing is that this complex cell doesn't care whether you present the bright vertical bar here, over on the left, or way over here on the right. So the complex cell no longer cares about the position of the bar in its receptive field. It's insensitive to the position of the bar. And that indicates that we are combining nonlinearly these simple cell inputs. But it is, once again, sort of the next stage of visual processing. Sometimes we want to know exactly where a bar is in a receptive field. If we do, well, we'll probably use a simple cell. But sometimes we just want to know that there is a bar somewhere in the receptive field. And if that's the case, we'll be interested in the complex cells. It turns out there are also hypercomplex cells and a large number of other sorts of cells up there in visual cortex. But this is a good example. So, we should understand now that in visual cortex we have cells sensitive to bars and to edges. They have different size sensitivities, so they could be looking for nice wide bars or very narrow bars. They also have different orientation selectivities, horizontal, vertical, tilted, etc. So what we want to do is now use such cells with the receptive fields that we've talked about to now try to detect patterns. And so we're going to talk a little bit about pattern recognition. And the sort of model we're going to examine is called by the textbook a feature net. And this is a model of pattern recognition involving a network of detectors with feature detectors as initial processing elements. So, what is a feature detector? Well, here's an example. What about an orientation selective bar detector? And again, an orientation selective bar detector, if we were to show it a bright vertical bar, this would result in stimulation of photoreceptors. And if this bright vertical bar were in the correct position, we would get a strong response out of a vertically oriented simple cell. So how can we use such things to detect a square? And is effectively the question we're after, and when it comes to pattern recognition. So, visual stimulation is effectively some pattern of light, and in this diagram that I'm about to present, the visual stimulation enters our feature net system at the bottom of the diagram. What we're going to assume is that the first stage of visual processing by this feature net involves bar detectors, and what we're going to do is effectively apply these bar detectors to the visual stimulus and see what's there in terms of bars. Now, a second stage could combine the responses of bar detectors. If these bar detectors are chosen appropriately, well, we could now be forming angle detectors. So if I had a simple cell that's looking there and a simple cell that's looking there, and I combine their responses, I'm now looking for a corner over there. That's the idea. So in particular, this guy and that guy will make that corner. Let's see, this low one at the bottom right, and this, the second from the left. What is that? That's sort of a backwards L shape. So you see we can get angle detectors if we combine responses of bar detectors. That's the point. So you can now ask yourself, how do we get a square detector? Well, we combine the responses of the appropriate angle detectors. And if these angles are located at appropriate positions in visual space so that they sort of line up with one another, the edges, then yes, we must be looking at something like a square. And so this is a feature net that would help us detect a square pattern from the responses of simple oriented bar detectors. And in this case, we have a model where the response is determined by the incoming visual data. And in this case, we have a bottom-up style of data processing. The result that we get from this visual recognition network is driven by the data. It's working in a bottom-up fashion. The visual stimulation determines what's happening. Either there is a square in some location or there isn't. It is determined by the data. Now, let's go beyond squares and consider letters. Feature detectors, again, are going to provide the initial responses of visual processing. And now we're going to consider visual inputs that can be letters of the alphabet. In this case, we're showing the letter T. So feature detectors could be simple ones, like a vertical bar or a horizontal bar, but they could also be angles. They could also be curvy things, like that C shape over here. If we have T as the input to some set of visual feature detectors, then the horizontal bar detector at the appropriate location will be stimulated. Why? Because there's a horizontal bar at the top of the T. The vertical bar will be stimulated by the vertical bar inside that T. Likewise, these two angle deals are present inside the T stimulus. So again, thinking bottom up, yes, we will have excitation of the 4 feature detectors shown here on the right. Now, what about this feature detector, which is looking for that curvy C shape? Is it going to get any joy at all from that T? No. What about these diagonal bar detectors? Are they stimulated by that T? No. What about that angle? No. These guys are not stimulated by the T letter. They're going to have to wait for some other letter to show up to be excited. So you can imagine now, if we're going to try to detect letters, it makes sense for each letter to receive excitatory inputs from those feature detectors corresponding to features in the letter. So if we want to have a T letter detector, it should be excited by these fellows on the right, which are actually present inside the T visual stimulus. The T letter detector should not be excited by those feature detectors shown here on the left because they are not present inside the T visual stimulus. I mean, that curvy C shape, well, it is present inside the C visual stimulus, and so it should contribute to a C letter detector, but, well, that's not our example. We're looking at the letter T. So the idea then is that features inside of a given visual stimulus, inside of a given letter, contribute to letter detector activation, and we choose the letter, the node, the letter detector with the greatest activation as our best guess as to the letter that we're looking at, and that would be the letter T. So this is sort of a schematic, a sort of simple diagram of how you might try to build an optical character recognition system. And this would be a system whereby, well, if you have a book and you have a scanner of some sort and you want to turn that into a Microsoft Word document where you're getting the actual letters that are on the page, well, you've got to recognize those letters from the visual image provided by the scanner. And so you've got to do optical character recognition. In any event, Oliver Selfridge described this sort of model a long time ago and called it a pandemonium model. And this is its bottom-up portion, where visual stimulation is exciting feature detectors, in turn exciting letter detectors, in turn leading to a decision based on a simple rule: which of our detectors has the greatest activation? In our textbook, you will find this, and I didn't find this as informative as the previous diagram, but again, the notion is that certain features are present in each letter, and so it makes sense to take inputs from appropriate feature detectors for each letter detector. And in fact, if you can combine the inputs of letter detectors appropriately, you should be able to build a word detector. And if you were to take appropriate word detectors, you should come up with sentence detectors. Oh, that might be one step too far. In any event, you get these hierarchical models where you can try to detect more and more based on an initial feature detector stage. Top-down processing, well, what does that say? The cat. And it's obviously the cat despite the fact that this and that are identical shapes. We see it as an H, T-H-E. We see it as an A, C-A-T. And we see it as an H or an A depending upon the context provided by the other letters. So the visual stimulation pattern provided by the guy on the left and the guy on the right are identical. So there's no amount of bottom-up processing that will make this one an H and that one an A, because the visual stimulation is identical in both cases. So it must be that there is some kind of top-down effect. And in this case, we say, well, yeah, it's the surrounding letters. That's a T and that's an E. What could this be but an H? C, T, what could that be but an A? So it's sort of our knowledge of words in the English language that lets us interpret this correctly is H and A. So that's top-down. So if we are looking, if we are reading a word and we are thinking about an animal, then this might affect our processing of that word we are reading. If we're thinking about an animal, then it might make us more likely to perceive the word cat or to perceive the word rat. It would make us less likely to perceive the word mat. So if we believe that the word that we're about to encounter is an animal, we will effectively prime certain word detectors, cat and rat, and we will inhibit other word detectors, in this example, mat. If we now have a bottom-up input from the visual stimulus, and we're looking at our first letter, well, this particular detector, which matches closely the C shape, will be activated. This will lead to excitation of the C letter detector, but not the R letter detector, and this will cause us to find greater activation of the cat word node than the rat word node, etc. So we have both top-down and bottom-up components of the model working simultaneously in this case. Okay, now what about form perception? We're going to look at Gestalt perceptual theory now, and the basic notion is that sensory features are grouped in a way that allows a figure to be perceived. And here's a nice example. Here we have a triangle, and you can see that it has these small constituents that are the letter T on the right. Horizontal line segments close to the right, dots close to the left, and then line segments simply over on the left. It's still the same shape. It's still the same figure. It's still that same triangle. And so then the question is, well, despite this transposition, we have the same figure. How can we account for that? How is it that we arrive at this sort of global shape or figure sort of in spite of changes in the features that can lead to it? So the first question you might ask, well, what are the sorts of features to which the visual system is sensitive? And I want to review briefly two ways that people have used to try to establish important visual features. And one of these ways is to do visual search experiments. And Ann Treisman did groundbreaking research in determining which visual features are perhaps the most salient or important when processing visual information. And in her experiments, she showed how a target shape or color or some target visual stimulus must differ from distracting visual stimuli in order for it to be detected easily. So let me give you an example. Let's suppose that we are told that this red O is the target, and our job is to find that target. Is it easy? It's a piece of cake. In this particular example, we find that red O instantly. In fact, it doesn't take any work to find the red O finds us. It more or less pops out of this display. In this display, well, you can sort of see why it pops out. Even though all of these visual stimuli have that same O shape, only our target has that red color. And so there is a color feature that allows the target to be detected when embedded among this set of distractors. And so we have our red target, our green distractors, and apparently that color feature suffices to distinguish a target very easily. So this guy ends up popping out, the red O, by virtue of different color. Did it pop out again? Was it easy to see that red O? Was your attention drawn to that red O? I think for most people it was. It popped out again. Was it by virtue of its red color? No, it was by virtue of its shape. And in this case, the O shape is very different from the V shape. So did it pop out that time? No. It might be easy to find, but it probably did not pop out, because in this case, there are distracting elements with the same shape, namely those green O's. There are also distracting elements with the same color, namely the red V's. So there is no single feature that allows the target to pop out. So that results in a harder search. So people sort of say, well, how difficult is search? They play around with the properties of the target and the distractors, and they eventually learn that things like, well, a difference in color, that can provide an important feature. A difference in shape, that's another feature. And you can come up with a list of features that make searches of this sort easy. Texture segregation experiments have also provided evidence for important visual features. And so there's a fellow named Bela Ulesh who did texture segregation experiments early on with stimuli sort of like this. Can everybody see what's going on the left side here? Most people would say yes. It's pretty easy to tell that there is that embedded square, and that square is different from this surrounding zone. Does everybody agree that you can see the square pretty easily on the left? Good. It's a little bit harder to see that square on the right. Do people agree with that one? There is a square on the right, just like there is on the left. This is hard to see. So on the right, it turns out you have plus signs in the middle, and then you have these sort of L's or partial plus signs that only cross at their, towards the ends in the surround. So there is a square here, but it turns out those perfect plus signs are different from those distorted L's, not enough to make an easily segregated texture. The plus signs are very different, though, from the L's, and that does make for an easily segregated texture. Here's another example. Texture segregation, we can clearly see the vertical edge between the L's and the T's, and this vertical edge between the T's and the tilted T's. And we can sort of see the sort of featural differences. People pay attention to things called T-junction. A T-junction is simply a junction that makes a T shape. There are no T-junctions on the left. There are T-junctions in the middle. So you'd say, aha, T-junctions may well be a feature that lets us segregate textures easily. We happen to know that orientation is a feature that lets us segregate textures easily. So if everything is upright on one side and tilted on the other, then we can easily see the boundary between upright and tilted because of the difference in the orientation feature. So that would account for this sort of thing here. Orientation is a feature. What are some more features? Well, up at the top here, we can see the difference pretty clearly between left and right, and we see that the stripes are bigger on the left and smaller on the right. And that's true also in the middle. They're bigger on the left and smaller on the right. And it seems as though stripe size may be one of these visual features to which we're very sensitive. It turns out stripe orientation, yet another thing, I think we mentioned this already, horizontal, vertical, there's a clear boundary between the two sides. So perceptual experiments of this sort, in addition to the visual search experiments, in addition to work in which the sensitivities of neurons in the visual system are measured, all provide information concerning basic features used by the visual system in processing visual data. And the features that have been identified are variations in brightness, color, orientation of lines, bars, and edges, scale or size, which is very similar to something called spatial frequency, lengths, curvatures, the way things move is very salient. If certain things are moving over to the right and other things are moving to the left, you'll be able to tell the difference instantly. Motion is a very important feature. So, now that we know what some of the features are, what can we do with them? Well, it turns out we can group them together to give us a figure. These figures are sometimes known as gestalts. And these gestalts can have what are known as emergent properties. So what's an example of an emergent property? Well, let's take component A here on the left, which is an L. Add to it component B here in the middle, that diagonal line segment. Put them together to get this shape, this triangle. And we now have an emergent property present in that triangle, but not in those two components. And that is the fact that the triangle is a closed shape. So this closure is something that emerges from the composition of those two elements, the creation of the triangle. Here's another example. Second row, we have that little line segment, and we're going to add it to this picture where we appear to have two squares. And there we go. And it now looks as though we definitely have two squares that vary in depth, and there is a horizontal surface that joins those two squares. So that horizontal surface emerges when we combine those features. Likewise, three line segments, single vertex, a hexagon, and if we now put them together, we have a three-dimensional shape with volume. So that is an emergent property. Seeing a cube is something that emerges from these simpler flat shapes. Convexity, that's not as strong an example. So, we get these figures or gestalts, they can have emergent properties, and it's important to note that a figure should be contrasted with a ground or the background against which the figure is seen. And this is perhaps best seen by rare examples in which we have reversible figures. How many people have seen this stimulus before? Yeah, this is one you'll see all over the place. This is what's known as a reversible figure. And I think when most people first look at it, they see some cup or vase thing in the middle, this bright shape. And after a minute, though, you realize that this is actually two people facing each other Here's the person on the left looking at the person on the right. And they're now in black. And what you have done is to effectively reverse the role of figure and grounds when you get those two perceptions. So when you see this vase in the middle here, the vase is the figure, and that surrounding black business is the ground that you're not paying attention to. However, if you stare at it for a bit, you can now see that the black has arranged itself into two faces. That's the new figure. What's this space in the middle? Nothing. That's just the ground. Okay? So that's known as a reversible figure. And it shows clearly the role of figure and ground. There are more reversible figures. I can look at this as a bunch of black arrows pointing upwards, in which case I do not see the white arrows pointing downwards, because if I'm seeing black arrows pointing upwards, the white arrows are ground. It's very hard to see both sets of arrows simultaneously in this diagram. If I see the black ones, the white ones just sort of merge into the ground. If I'm looking at the white ones, then the black ones are ground. How many people see that? Good. It's sort of hard to see them both as figure. Here, it's very easy to see the white ones as the figure, with the black area acting as ground, but it is possible, of course, to see the black arrows as well, with the white ground. And finally, most people find it easy to see black waves, so that's the figure, and this white business would be the ground. It turns out that you can also see these white guys. It's just a little bit harder, though. Then the black is the ground. Which is easier to see here, the white or the black? I think it's easier to see the white, just like it was easier to see the black over here. And people note that sometimes with the reversible figure there is a bias to see one figure rather than another. And what would the bias be in this case? Well, perhaps gravity, who knows? Certainly the black is preferred here, looks like a wave, and the white is preferred over there. What accounts for that? It's hard to say. So the visual system is clearly trying to make sense of what it's looking at. It's organizing smaller parts of the visual world to create more complex wholes. And this activity of the visual system is shown pretty clearly by what you perceive when you look at patterns of this sort. This is known as a glass pattern, and most people perceive it as very, very busy. How many people see this as a busy pattern? Why is it busy? Well, there are these circles that are constantly reorganizing, sort of moving around, and some will come up and then they'll go away and new ones will come up. There are no actual circles drawn in this picture. There's a whole bunch of dots of different sizes and various shapes. There are no real circles that are drawn. But the visual system is working hard to interpret what it sees, to put together features to create greater levels of meaning. And in this case, well, it's working very hard, and it sees all these circles that are, in some sense, are illusory. Okay. So how do features get organized to produce figures? There are perceptual organization laws promulgated by the Gestalt psychologist, of which Max Wertheimer, of whom Max Wertheimer is perhaps a leading proponent. And this is way back almost 100 years ago now. We're going to go through these laws with visual examples, but I will note that a lot of the Gestalt laws of perceptual organization apply also to other modalities, such as hearing, somatosensation, the other senses, in effect. But we'll look at vision examples. And the first of these laws is proximity. Things close to one another get grouped together. So it's frequently the case that these people are sitting beside one another and I say, well, I'm going to group them together. And it turns out that that is a good thing to do because here are the TAs for for the most part, except for that one over there. So I've grouped together these students who sit together. Now I also see that group there, and I've seen that group there before. You know, there are various groups of students. They're close to one another, and I sort of note that maybe they know each other and they're friends or something. That is an example of something that's more general. Things that are close to one another get grouped together. So here we have these 6 spots. But most people would say, well, it's not so much 6 spots as it is 3 pairs of spots. The pair on the left, the pair in the middle, the pair on the right. And so when we think of it that way, when we see it that way, it's clear that we are using the law of proximity to group things that are close together. These 2 guys are close to one another, so they group together as a pair. Ditto with the 2 in the middle and the 2 on the right. Notice that it is perfectly possible for the grouping to have worked in a different way. It could be that we would take this second one from the left and associate it with the third one from the left. So these 2 get grouped together. We could then group together the 4th and the 5th. Finally, we could, and I'm saying we could do this, but we don't, we could group the 6th and the 1st. It's actually very, very hard to see this particular stimulus that way, with the 2nd and the 3rd, the 4th and the 5th, and the 6th and the 1st grouped together. What is very easy is to group together the ones that are close, 1 and 2, 3 and 4, 5 and 6. So that's the law of proximity. Similarity, things that are similar to one another get grouped together. And here we have an example where similar color determines grouping. So on the left and on the right, we have 25 dots. The dots are all of the same shape. The only thing that differs is the color. And on the left, we've got a row of red, a row of green, red, green, red. And when we look at the thing on the left, well, we say we have 5 rows of dots. When we look at the right, we have a column of red, a column of green, red, green, red. And when we look at this stimulus, we say we have 5 columns of dots. In fact, it takes a little bit of work to see this one on the left as 5 columns. It's very easy to see 5 rows of dots. It's hard to see 5 columns. You almost have to force yourself to see 5 columns. And this one on the right, it's very easy to see 5 columns, much tougher to see it as 5 rows. Clearly, we're organizing things according to color similarity. Similar feature. Now, color is just one feature. It could also be brightness or orientation or another of these features that we made a list of earlier. As long as they're similar to one another, they'll be more likely to be grouped together. Our example, it turns out to be color similarity, but there are other things that can be similar. So, proximity, similarity, closure. When we look at this thing here, well, duh, it's a triangle. That's the way it looks. But of course, it's not a triangle. It's a couple of line segments with a common vertex at the top. And it's this set of 3 line segments with 2 vertices at the bottom. That is not a triangle, but we do see a triangle nonetheless. And this property of joining the top part and the bottom part, in this example, is known as closure. I see a hand.
Just out of curiosity, about what age in a child would they start to recognize that? Is it grade school?
Oh, by 2, certainly.
By 2?
Yeah. We have a developmental psychologist in the Department of Cognitive Sciences, Barbara Sarnica. Anybody taken a course with Barbara Sarnica on development? In any event, I can definitely recommend that sort of thing. Edge segments will be joined if joining results in a closed figure. So, in other words, these 2 segments on the left, they line up with one another. And if you simply continue them towards one another, you'll see, yes, they line up with one another perfectly. This is not something that occurs very easily just by chance, just by accident. there's probably some reason in the world that it accounts for these two line segments lining up so perfectly. So the reasoning goes. Similarly on the right, this thing on the top lines up perfectly with that thing on the bottom. Is that an accident? I doubt it. There's probably some reason. And what is the reason? Well, the reasonable interpretation is in terms of occlusion. And occlusion is the same thing as interposition. And effectively, both terms refer to something in front blocking whatever's going on behind. And in this case, the something in front might be a bar that is blocking the edge on the left and the edge on the right. Okay? So if there is an interpretation like this in terms of occlusion, then we are more likely to close up those gaps here and to see a closed figure. And that's closure. Good continuation. Well, when we look at a stimulus like this, which is this curvy X thing, it looks basically as though there's one curve going from top left to the bottom right, and a second curve going from the top right down to the bottom left. And that interpretation is not only the one that I think almost everybody sees, it's also one of several possible interpretations. So for example, it could be that what we're looking at is this sort of pointy thing starting at the top left, going to the central vertex, and then going back to the bottom left, like this blue thing shown at the bottom. Or we could be seeing this guy at the top right, going to the central vertex and down to the bottom right, like this red one. You know, how many people see this when they look at that thing up there? Almost nobody sees that. I think almost everybody says, well, there's something going from top left to bottom right, and something from top right to bottom left. They almost never see this or some other crazy interpretation. So people have looked at a lot of stimuli like this, and what they said is, well, there must be some reason for this, and the reason is good continuation. And the easiest way I think of it is when you're driving a car and you come to an intersection. So let's say start at the top left, I'm driving a car, zoom, 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 and I come to an intersection. What would I rather do? Continue going straight, or would I rather stamp on my brakes and pull a sharp turn? What's the easiest thing to do when you come to an intersection? Continue going in the same direction you have been going. Continue the same way. Simply drive through that intersection without turning. So that's good continuation. So we're driving along, we come to the intersection, tough, we keep on going along. What about this? We start here, here's the intersection, tough, we keep on going. Something like this does not conform to that good continuation rule. There's not a smooth continuation in this case. Rather, there's a sharp turn at this vertex and another sharp turn at that vertex. So that's not good continuation. So you continue an edge in a way that minimizes change in direction. That's one formal way to say it. Common fate. I don't think this appears in your textbook, and I'm not certain why. Gestalt law of perceptual organization, there's a number of them, and one of the most important is known as common fate. Things that move together get grouped together. So you can see we have this set of purple dots. Some of them are moving one way, some of them are moving the other way. And it's very clear that we can see those two groups of dots based on their different motions. So things that move together get grouped together, and that's known as common fate. So that's a good grouping principle. Then we begin to get things that I don't understand as well, but I will mention them. The law of pregnance. Organization of the visual ray into perceived objects will be as good as prevailing conditions allow. What is meant by good is regularity, simplicity, and symmetry. The simplest interpretation of this figure uses closure, and when I just don't understand what this is, to tell you the truth. So the law of pregnance is apparently a law of perceptual organization. I never understood it And so I'm not even going to try to explain it. Perhaps one of you can come to office hours and explain it to me. In any event, I do mention it though. Now, what about this? One of the things visual perceptual psychologists love to study are contours. And there are two kinds of contours. There are contours that belong to an object or a figure. These are known as intrinsic contours. And then there are contours that are a consequence of interposition or caused by some object being in front of the figure of interest. And these are known as extrinsic contours. So it turns out the contours can be filled in, particularly the extrinsic ones. And here's a really famous example, Kinesa triangles. So if we look at this stimulus on the left, what do we see? This little shape is called a Pac-Man. People remember the Pac-Man game? Okay, so that's called a Pac-Man shape. So we have these three Pac-Men, and we also have this little pair of line segments on the left, this little pair of line segments on the right, and that pair of line segments at the bottom. And those are really the only things in that picture on the left. The three Pac-Men and the three partial triangles. But what do we actually end up perceiving? Something that's a little bit richer, isn't it? First of all, I think most people would agree that we see a white triangle in front. Yes. And what kind of triangle do we see behind that white triangle? Well, we see the triangle with the black edges that is created through a process of closure. Neither of those triangles is actually in this picture, of course, but we perceive them very clearly. And it turns out that what we are doing is filling in contours, particularly extrinsic contours. In the case of this triangle that lies behind the white one in front. So the one in front, we are seeing subjective contours. And those are the contours that we see as edges for this triangle right here. So if we look over here, now on the right, we see a similar figure. We're no longer using filled-in Pac-Men. Rather, we have these sort of circle sectors. But we see a similar organization. We have a white triangle in front, and it has effectively subjective contours for its edges. We see the triangle in back with its black edges, and its contours have been filled in where it's blocked. And these are pretty neat. So we have intrinsic contours belonging to an object or figure. For instance, the boundaries of these Pac-Men, the circular boundaries here. Extrinsic contours caused by interposition. We talked about those. Those are the ones that get filled in. And finally, subjective contours, like the contours surrounding these white triangles that lie in front of such a figure. We have a specialist in just this topic, Professor Don Hoffman. And Don has a web page where you can get more of this. It turns out that contours may be filled in other ways, and one example is by neon color spreading. And so let's look at these one after the other. The neon disk, we're going to start out with just these red segments, and then we're going to extend them using those black segments. And when we extend those red segments with the black segments, you should be able to see that the red sort of bleeds into the surrounding area to give you a red disk. So rather than seeing just red line segments, as we do here, now we should see a red disk. There's sort of this shimmering neon redness making a disk when the black is present. And the same thing with this worm. If we're looking just at the blue as right here, then we don't see anything special. But when we add the black segments, we now get this two-dimensional filling in. So we see this blue band, the neon color spreading. When we have these little dots here, there's no neon color spreading, but now we see actually this sort of blue neon square hovering in front of the figure. So those are further examples of filling in. And at this point, I want to say this. I think we're just about done. As per the textbook, similarity, we tend to group these dots into columns rather than rows. Things that are similar to one another get grouped together. Proximity, we tend to perceive groups linking dots that are close together. Things that are close to one another get grouped together. Good continuation, we tend to see a continuous green bar rather than two smaller rectangles. We tend to continue things in the easiest possible direction, as similar as possible to the original direction. Closure. We tend to close boundaries, particularly if there is an interpretation that relies on occlusion or interposition by some object in front. And finally, simplicity, and I'm not certain what that means. It's part of the law of pregnance, and it's fine. If you were asked for an example of 5 Gestalt laws of perceptual organization, in my mind, you would dip this one, get rid of this one and add common fate instead, because that's a very nice little grouping principle. Things that move together get grouped together. So that's it for today. We will come back on Thursday and continue Chapter 5. Have a good one.